today we have an exciting lecture also. The title is Developing, Validating, and Implementing Liquid Biopsy Techniques or Technologies for Pediatric Solid Tumors. And we have a great friend to the foundation, Brian Crompton, who's the research co-director at the Solid Tumor Center up in Boston, assistant professor of pediatrics at Harvard Medical School. And from his lab's website, I guess their mission statement is harnessing novel genomic and proteomic approaches to improve outcomes in pediatric solid tumors. So with that, I will turn it over to Brian. Thank you for coming here today and presenting this lecture. We appreciate it. And I'll turn it over to you now.
Hi, everybody. It's really great to be here. I think that's the first time anybody's actually read my website, maybe. You need the grandiose, ambitious statement on your website. So, and we are ambitious. We are going to try to make things different and better for pediatric patients with cancer. And one of the focuses in the lab, and really our biggest focus now, is really developing and adapting and validating and implementing liquid biopsies. So it's really exciting to be here. I'm looking forward actually the most to the Q&A session. And, you know, I think the important background here is that this is a work in progress. There are an extraordinary number of people who are contributing to trying to make this new technology useful for caring for our patients. And that extends from the groups in the labs beyond our lab. There are multiple groups working on this. The clinical investigators who make sure we get access to samples. This is a lot of work to do that. And writing a trial is no small feat, as everyone knows to begin with. And then most importantly, patients and families who agree to contribute a sample for research purposes when they're enrolled in a trial. We can't do this work at all without them. Just want to make sure, Jay, am I showing the right, are you seeing the full screen?
Yes, we are.
Great. All right, so let's jump into it. So liquid biopsies are this concept that you can find fragments of tumor material in a blood sample. It's not just blood. We also can find fragments of tumor material in a pleural effusion, a cardiac effusion, in the cerebrospinal fluid in patients who have brain tumors or spinal tumors, and even in the urine. So the reason we mostly focus on blood is quite frankly because we get a lot of blood samples from patients who are undergoing treatment for cancer. And so it's the most natural place to look. And you know, the other advantage from blood is that the blood system's everywhere. And so wherever there's a tumor, there's a blood supply. And so there's a reasonable chance that that blood supply will pick up fragments of tumor wherever a tumor is. And there are different types of fragments that end up in the blood. We're going to spend most of the talk focusing on circulating tumor DNA. But the real reason for that is that that technology is just way further along than everything else. But there are many other things in the blood that can be shed from a tumor. We also will spend a little time at the very end of the talk, if we have time today, talking about circulating tumor cells. But there are exosomes, cell-free RNA, all kinds of things. And of course, you know, there's a long history in the care of patients with cancer of following tumor antigens like the PSA test in prostate cancer. So this is sort of a new field embedded within a well-established field within adult and pediatric cancer. So we're going to start by talking about cell-free DNA. And one of the most important aspects of this is to keep in mind that lots of cells in our body, especially the normal white blood cells that circulate in our blood, will shed cell-free DNA into the blood. And so we think where cell-free DNA comes from is that cells eventually get old, break down, or outstrip their blood supply, or are programmed to die. And as they do so, they shed this DNA as the cells rupture into the blood. And it doesn't stay there very long, but there's sort of a constant trickle of cell-free DNA in the blood that's getting cleared out. And for some reason, it seems like tumors, especially tumors that are highly metastatic and are very aggressive, seem to shed a reasonably high level of cell-free DNA. It's definitely disease specific how much that shed is occurring. But because normal cells shed cell-free DNA, you can't just look to see how much cell-free DNA is in the blood to figure out how much circulating tumor DNA is in the blood. And that's really important because there are kits out there that you can buy from QIAGEN, for example, that will take a plasma sample, extract the cell-free DNA, and then of course it's simple to quantify how much DNA you get from an extraction kit. So for that simple, we'd be much further along. But of course, because regular normal cells also shed cell-free DNA, we need to have a way to differentiate circulating tumor DNA from cell-free DNA to know that what we're finding is really originating from the tumor. Just to show that we're going to get a little more into how we do that, but just to show that it's feasible to find circulating tumor DNA across different cancer types. This is a plot from some older data, mostly in adult cancers from 2014, from Luis Diaz's group. Was one of sort of the hallmark liquid biopsy studies that came out several years ago. And then a plot from our lab from 2018, where we just show that circulating tumor DNA is present in most of the common adult and pediatric solid tumor cancer types. In my lab, we haven't really focused too much on brain tumors, but it's clear that from in brain tumors, there's limited shed into the blood, but probably a reasonable amount of shed into the into the CSF that bathes the brain and the spinal cord. At least for some subsets of brain tumors. And so that's also a source of circulating tumor DNA. So as I mentioned, the trick with detecting and quantifying circulating tumor DNA and really profiling it is to really be able to figure out whether you have tumor DNA or whether it's just cell-free DNA from normal cells. And you can't just assume that it's there because the patient has cancer. Some patients with small tumors really shed so little it's very hard to find. And so the way we go about finding circulating tumor DNA is to find fragments of tumor DNA, of cell-free DNA, that have the mutations that characterize the patient's tumor. And so we all know that mutations are one of the things that make a cancer cell different from a patient's normal cells in their body. And so if we can identify these mutations, we can be sure that what we're finding is circulating tumor DNA and not just cell-free DNA. But a lot of the technologies that were initially developed for this were really hyper-focused on point mutations that are very classic drivers of adult carcinomas like colon cancer and breast cancer, et cetera. And we know now from a lot of work from a lot of really great scientists in pediatric oncology that the mutations that characterize pediatric solid tumors are different than those adult carcinomas. And what we see more and more often in pediatric cancers are structural variants, so translocations or copy number events. And those are different. You need different sequencing and profiling technologies to find those in the DNA than you do to find a point mutation that activates a tyrosine kinase. There certainly are activating mutations, these single nucleotide variants in pediatric tumors, but even in embryonal rhabdomyosarcoma and neuroblastoma, where they're relatively frequently found, they only occur in these recurrent variants in about half of cases, or sometimes even just a third or a quarter of cases within a disease, as in neuroblastoma. So we can't rely on that, because if you don't find a point mutation, you can't assume that there's no tumor DNA there, because it may be that that patient's tumor doesn't have a point mutation in one of the genes that are commonly mutated in a pediatric cancer, or commonly looked at as a cancer gene. So we really need to to make sure we're really finding circulating tumor DNA. We've really taken the approach that we need to be able to at least capture copy number variants and translocations that are relevant to the diseases we study. And then, of course, we're also interested in finding the point mutations, but we're not relying solely on that to find the presence of ctDNA. And so the other sort of thematic approach that my group has taken is to not be also constrained by a very specific technology. I think there's a tendency, and we're certainly guilty of this, that when we develop an approach, it's exciting, especially when it works. But we try not to get too married to that approach, because it may not be the right fit in all scenarios. So up in the upper right panel, you'll see that we really push this approach of trying to use the detection of translocations to identify ctDNA and translocation-driven cancer. But of course, that doesn't work. Our NGS approach doesn't work if the tumor doesn't have a translocation, which would be obvious. But it also has limitations of sensitivity. So while it's really good for differentiating who has high levels of circulating tumor DNA and who has low levels of circulating tumor DNA, if we want to find the presence of minimally residual disease, meaning a patient looks from a scan to be cured, but we worry and suspect that there might be some residual cancer there that will eventually give rise to relapse, next generation sequencing approaches for detecting a fusion, even in a patient who has a fusion positive cancer type, is not going to be sensitive enough. And so my lab is really interested in utilizing whatever technology has been developed out there for circulating tumor DNA. And we really only focus on assay development when we see a gap that we really need filled for a disease we work on. That's partly why we developed a custom NGS panel for finding fusions. But we've adapted ultra-low passage whole genome sequencing, which has become really a workhorse for the lab for identifying patients with really high levels of ctDNA. And my lab didn't develop that. That was developed at the Broad Institution and by other groups. We use ddPCR when we really need high levels of sensitivity, and multiple other groups in the US and in Europe published studies before anything in my lab was published to find translocations and other variants. And then we do like sort of broad sequencing, and we partner with groups who do DNA modifications like methylation and 5-methylcytosine type of NGS studies that just tell us different things about the features of DNA that make the tumor DNA different from the normal. And then there's a lot of other exploratory technologies that we are working on, mostly with partner groups, often with computational biologists who sort of specialize in these new sort of computational technologies. So we have like this this toolbox of different approaches for detecting ctDNA, and they should help with different types of cancer types, but also help with different indications. So then the question is, what are the indications for detecting and profiling circulating tumor DNA? What is it good for? And there's several areas where, when you think about cancer as a field, where there's pretty good evidence that these technologies could be very useful, both from a patient care perspective and from a biologic discovery standpoint. And we also take the view in my lab, and I know probably everyone on this call has this view, that pediatrics should never be behind in leveraging these opportunities that we know are valuable for understanding cancer better. And so the ways that liquid biopsy is often used in the cancer field is at the time of diagnosis. There's really kind of two utilities there. One is if you're looking for a mutation in a gene that could potentially help you pick from one or multiple different therapeutic options, it can be used in a similar way to a biopsy sample to profile the patient's tumor and find out if, for example, there's an ALK mutation that suggests you should add an ALK inhibitor to your therapy. But we've also found that knowing how much ctDNA is there may help us do a better job of risk stratifying up front. And we know from a lot of the diseases we work in, especially neuroblastoma and Wilms tumor, that risk stratification can really help us improve outcomes and at the same time mitigate long-term toxicities that is commonly associated with the therapies we use to treat children. We think, as a pre-treatment time point, prognostication is possible, but also following how rapidly patients respond to therapy can also be prognostic and help us decide whether we think the therapy we're given is the right therapy for that patient's specific tumor. And so watching sort of on therapy response to treatment may be a really good use for ctDNA. That aspect of what we're doing still needs a lot more validation, but that's in the works. And then, we hope that we can use this to detect relapse early. And we know that, once relapse occurs, it's harder to treat. But we've really not ever had a fair chance, I would argue, in pediatric solid tumors to find relapse disease when it is really minuscule, barely there. We usually find it when a patient has symptoms and has, sort of bulky disease that is easily picked up on a radiograph. And I think that we need to revisit that when we're able to find relapse at a much earlier stage. And I have some hope, I think, that relapse therapy might be more effective and sometimes curative if we can employ it much earlier when we find that patients have minimal residual disease, even though we think that they should otherwise have been cured. And then in the context of relapse and progression, not only can it still be used to help us track how patients are responding to, sort of relapse therapies or clinical trial therapies, but we can also use them as a discovery tool, because I think it's clear now that tumors during the course of upfront therapy change over time, that there's a selection pressure where portions of a patient's cancer that's most amenable to therapy is pretty effectively treated by our upfront standard care. And what's left is what gives rise to relapse. And because those are the resistant cells, those resistant cells somehow are more refractory to second and third line treatments. So if we can understand how those tumors have evolved, we may be able to prevent the emergence of those relapse clones, or maybe even think about new therapies that are very specific for patients who have developed relapse or progression. Cell-free DNA is only going to tell us about how the DNA level changes that have occurred over time. There may be other ways with other liquid biopsy technologies to also think about understanding tumor evolution and disease heterogeneity. But one of the reasons why this is such an advantage, using liquid biopsy is such an advantage, is that despite best efforts over decades now, it's been really hard to get large collections of relapse biopsy tissues. And for good reason. Most children, when they relapse with cancer, a biopsy is not going to actually help us change therapy. And obviously there's ethical and moral concerns about doing a surgical biopsy under anesthesia, particularly, that is only going to serve research and not provide any potential benefit to the patient. But blood samples are minimal risk, and patients are frequently getting blood draws anyway. And so getting multiple time points during a patient's relapsed or progressive course is completely feasible. And I'll show you a little bit of what we're finding in that context, although it's very early days. So thinking about now this sort of prognostic utility of ctDNA, I'll take you back to one of our first clinically relevant studies that we published. We used that next generation sequencing panel that we developed that was specifically honed into the detection of translocations that define Ewing sarcoma and a few other translocation driven cancers. And we just found that patients with higher levels of ctDNA at the time of diagnosis clearly do worse, even in the context of a cohort of patients who only had localized disease. So these are not patients, this is not a mixed cohort from this study. We didn't have some patients with metastatic disease, some patients with localized disease. Everybody had localized Ewing sarcoma. And yet we could clearly see a difference in the amount of ctDNA that was shed at the time of diagnosis. And those with higher levels of ctDNA clearly were more likely to relapse. And then importantly, patients with very, very low levels seem to do better. So I think this, as we refine this and validate this with a prospective cohort, one of the ideas is to make sure that patients who look like they're still at very high risk of relapse on standard therapy are enrolled more regularly on high risk clinical trials, and patients with very low risk of relapsing after treatment could be even considered for trials that test out intensity reduction. And that's been successful in some diseases such as Wilms tumor and Hodgkin's lymphoma, where dose reduction has not worsened outcomes have stayed stable, but have really reduced significantly the toxicity of our therapies. And then we had a similar finding in osteosarcoma, where we used this low passage whole genome sequencing approach to find ctDNA by identifying the sort of crazy copy number changes that are ubiquitously associated with osteosarcoma, and again found that we could find patients who had high risk and lower risk of eventual relapse after therapy just by looking at the pretreatment ctDNA levels. So David Schulman here at Dana-Farber is leading a multi-institutional study to validate this, but importantly, a big focus of the study is to actually look at some of these early and late time points to see if those also can predict even better than pretreatment time points, whether patients are doing well on their therapy or likely to have already developed resistant disease. This study now has pretty much accrued all the patients that we need to meet our statistical endpoints. There are patients who enrolled recently, and so we're still collecting those serial blood samples on this study. And then we'll have to monitor outcomes for a year or two at the end of the study, but this project is well underway. So I wanted to also, though, make sure we spent some time today focusing on some news stories. I want to make sure that we're demonstrating that we're pushing this field forward and not just rehashing things we published a couple of years ago. So this is a study that just came out in JCO a few months ago, where for the first time for our group, we looked at the detection of circulating tumor DNA in a renal tumor. So this was a retrospective cohort of blood samples that were collected from patients with stage 3 and stage 4 Wilms tumor. And we really honestly didn't know what we'd find. They were not ideally collected samples. These were samples that had been collected as serum, which is not always ideal for liquid biopsy use. But they were collected at a time when liquid biopsy wasn't really a thing yet. So understandably, this is what we had in the COG repository, and that's what we went to and to start off the study. So these were 50 patients who had a tumor sample, a serum sample, and a urine sample, and we wanted to just ask the question: could we detect circulating tumor DNA in the blood and urine of patients with Wilms tumor? And we focused on copy number changes because, as I mentioned earlier, while a portion of Wilms tumor, a portion of patients' Wilms tumors have single nucleotide variants that can be drivers in this disease, more common, all Wilms tumors really have copy number events. And so this low passage whole genome sequencing approach really is able to tell us who has high levels of ctDNA and who doesn't by identifying these copy number variants. And then the other benefit of that approach is that, over the last 5+ years, many Wilms tumor investigators have shown that certain copy number events are highly associated with resistant disease. And in Europe, for example, patients are frequently treated before a biopsy is done. So this data they don't have access to when patients are initiated on therapy. And even in the US, there are cases where upfront resection or biopsy aren't always feasible because of the location of the disease or the amount of disease present. And so, we also wanted to ask the question, could we consider whether we could use liquid biopsy to find some of these prognostic variants that are well established now from a liquid biopsy in the context of a case where maybe biopsy tissue isn't otherwise available. And we were sort of disappointed to find that very few urine samples had enough ctDNA for this method to be useful for detecting the presence of ctDNA. There were a lot of caveats with that. The most important caveat of that effort in the urine was that the volumes were very low. Usually we had significantly less than an ml. And of course, we all know that, like, if you want to collect for a while, you can get bucket loads of urine from any patient. It's completely non-invasive to collect urine. So there's no reason why we should necessarily consider this case closed. I think we need to think about whether larger volumes of urine could give us the answers we need for Wilms tumor. But suffice it to say that for the urine we didn't detect enough ctDNA to really be very useful in this cohort. But we were able to detect ctDNA in the vast majority of patients in the serum. And that was really striking, actually, more striking than I would have anticipated given our prior work in sarcomas. And importantly, we were able to find copy number variants, including 1Q gain, 1P loss, and 16Q loss, which are well validated prognostic mutations in Wilms tumor, all of which make patients' disease higher risk for eventually developing relapse on standard therapy. The other thing that I'll draw your attention to on the right, I hope you can see my pointer, is that when you match up what we saw in the tumor with what we saw in the blood and the urine, mostly it matches really nicely, but there's actually this group of patients where we found 1Q gain, which is probably the strongest prognostic factor of the three, in the blood where we didn't detect it in the tumor. And there's another study that recently came out in the context of patients with pre-treatment biopsies and metastatic biopsies from Elizabeth Perlman's group that shows, this may be in press actually, that shows that 1Q gain is often seen in the relapse when it wasn't seen in the initial biopsy. If this holds up in future validations, this would suggest that the 1Q gain is there. It's just present in subclonal amounts, so that a biopsy may miss the presence of 1Q gain. But in that subset of cells where 1Q gain is present, those are the cells that are more likely to give rise to relapse. And if we knew that up front, we might have considered intensifying therapy for these patients. And so knowing whether 1Q gain is there or not may be more at all, maybe more discoverable using a liquid biopsy over than a biopsy sample that's sort of geographically constrained to a certain part of a patient's tumor. We also found that we could find point mutations. When point mutations exist in the tumor, we were able to find them mostly in serum samples, occasionally in urine samples when ctDNA was present. And there were even a couple of cases where, a variant was found in the tumor, but not in the liquid biopsy, and even vice versa. So that, again, sort of speaks to the need to sort of capture heterogeneity, I think, in our pediatric cancer populations to really make sure we're giving them the right therapy. And then on the right, we were anticipating being underpowered to really show whether ctDNA detection in this cohort was prognostic or not. In part, that's because Wilms tumor patients generally do quite well, and so we knew the number of events from a cohort of patients of 50 patients would be rather small, and it's a small cohort to start with. And it's true that we didn't see a significant difference in outcome, but it's also notable that really all the events, all the relapses and eventual deaths in patients in this cohort, all of them had ctDNA detected at the time of diagnosis, and none of the patients who had very low levels, below the level of detection for our assay, ever experienced a relapse or death from disease. This obviously has to be validated. It's not significant. No one should change therapy based on this knowledge yet, but it is very exciting to have something now that can be a hypothesis for prospective ctDNA work. This is another new study that is not published yet, but we're really far down the tracks of trying to get it published, hopefully. This is a study in intermediate risk rhabdomyosarcoma. We were very interested in intermediate risk rhabdo because in some ways, intermediate risk is sort of like a middle bucket of patients where, outcome is expected to be in the 50 to 60% range, which, it doesn't mean that all patients do 60% good or bad. It means, that there are these patients who are resistant to standard therapy and patients who do very well with standard therapy. But it's a group of patients that we don't know how to further differentiate into high risk or low risk, because obviously it'd be better to do that. Then we would know exactly who should be treated with standard therapy and who should go on to newer agents in the context of clinical trials. And so we wanted to ask the question, could ctDNA help us here? We also wanted to just ask the question, because this study took many years to develop. We weren't even sure how often we'd be able to detect circulating tumor DNA in patients with rhabdomyosarcoma. So we wanted to just ask the question, can we detect circulating tumor DNA? Based on some of the prior work that I just showed you, we thought that we should try to capture as many of the variants as possible. As people here in this group probably know, rhabdomyosarcoma sort of has two genomic phenotypes. There's the embryonal, so they have histological phenotypes. They have the embryonal and the alveolar, and within those, most of the embryonal phenotype are fusion negative, but have RAS pathway mutations and are ubiquitously express copy number changes, whereas the alveolar rhabdo are mostly fusion positive, PAX3 or PAX7 FOXO fusion translocated tumors that rarely have other point mutations and may have actually very few copy number variants as well, because they're, the really main oncogenic event is this translocation. So we took an approach of using everything we could think of to try to detect circulating tumor DNA, and what ended up working the best for the fusion negative rhabdomyosarcoma was our low passage whole genome sequencing, because it could detect copy number variants regardless of whether there's a point mutation in the tumor or not in one of the recurrent variants we were looking at. And then for the fusion positive was finding the translocation was the most useful. But we applied everything to both cohorts. And what we found is that when we looked on the left here is a plot with open circles showing when we found a point mutation in a tumor, and the red dots are the same point mutation in the blood sample. Again, we were using serum because that had been what was collected in the COG studies. And what we found is that when ctDNA levels were reasonably high at a high enough level to be to detect the ctDNA, we were able to find the same variant. But in some patients, we found point mutations in tumors, and we weren't able to detect any ctDNA. So of course, we weren't able to find the variant in their blood. In some interesting cases, we were able to find some point mutations like a PIK3CA mutation only in the serum that wasn't previously detected in the tumor, and some of those were at very low allelic frequencies, suggesting to us that these were probably again heterogeneous events that were present only in a subclonal population of tumor cells. But I think so this was very exciting, very interesting. There's a reason why I think finding some of the point mutations in this disease are super important. I'll mention that in a second. But then what we did was we tried to differentiate high versus low. And since use of point mutations was only applicable to a subset of our embryonal rhabdo patients, but detection of copy number changes was applicable, copy number changes were present in all the embryonal tumors, we focused on detecting ctDNA by our ultra-low passage approach for risk stratification. And we found that patients with high ctDNA again did much worse, significantly worse. And in this context, these Kaplan-Meier curves are like screaming significant. We're more likely to relapse and more likely to die of disease if ctDNA was detectable than patients without. And we think of intermediate risk rhabdomyosarcoma as being sort of a 50 to 60% risk stratification. You can see that the patients who had higher levels of ctDNA had outcomes in the 30% range, whereas patients who had very low levels were more in the 75 to 80% range. So this could really help us think about redistributing the intermediate risk rhabdo cohort into high and low risk. But what was really interesting about this specific cohort is there was a recent study that showed that MYOD1 mutations were extremely poorly prognostic for embryonal rhabdomyosarcoma. And 3 of our cases had, both in the tumor and the plasma, detectable this MYOD1 mutation. What was interesting is that the patients with MYOD1 had very low levels of circulating tumor DNA, so low that they weren't detected at all by the low passage whole genome approach. And the reason that I think that this is so fascinating and so important for us to be cognizant of is that I don't think that we should be thinking about liquid biopsy as a one-size-fits-all solution for prognostication. We need to be thinking about integrated approaches. And so there are clinical factors that are still going to drive risk stratification. Of course, the presence of metastasis is uniformly higher risk in pediatric solid tumors. But MYOD1 seems to be a stronger prognostic factor, but only relevant to a very small portion of our patients than circulating tumor DNA. And then after you take those patients out who had MYOD1, for the remainder, the bulk of the patients in our cohort that had embryonal rhabdo, ctDNA remains the best next clinically prognostic variant. We found very similar findings for the fusion positive. One interesting finding is that, because we're looking at a DNA level, all the breakpoints for the translocations we found are in the intronic regions, even though the same exons come together over and over again for PAX3 and PAX7 FOXO rearrangements. The DNA breakpoints can be different, and we've actually saw what has been previously published decades ago, that the DNA level breakpoint, we never found the same DNA level breakpoint for the same patient in both genes over and over again. So everybody had a unique DNA level breakpoint for their PAX3 or PAX7 FOXO fusion in their tumor. And again, high levels of ctDNA versus low levels of ctDNA was prognostic. And then we took the overall cohort and we put it together, and we found, again, that for all of patients with intermediate risk rhabdo, regardless of the disease type, that detection was highly associated with a worse outcome, very significant, whereas lower levels of ctDNA were associated with a lower risk of relapse and a lower risk of death. We plugged this back into a multivariable analysis that took into account a whole number of other potential clinical variables that people have thought about as potentially prognostic. Of course, we knew that they're not prognostic because otherwise those clinical features would already have moved a patient in or out of this intermediate risk cohort. But we looked to see in this subset whether patients with ctDNA detection whether it remained a good prognostic variable, even in the context of all these other clinical variables. And in every way we've done now, in part to respond to some of the reviewers' comments for this study, we've done 4 different ways of this multivariable analysis. And for this overall cohort, ctDNA continues to be significantly associated with outcome in a multivariable analysis with the highest hazard risk ratio, both for EFS and OS. But one of the other factors, the only other factor that ever pops up is invasion. And while we know that invasion is really useful for prognostication, with more and more patients who have rhabdomyosarcoma getting delayed excision, we can't really evaluate how invasive the tumor is under the microscope until potentially months after they've started therapy. So we really think this pre-treatment ctDNA value is going to be really useful for rethinking risk stratification. We clearly have to validate this prospectively before we start making clinical decisions on that. But we're doing that in the context of the now closed intermediate risk rhabdo COG study. It's closed to accrual because it met its accrual goals, and now we have several years to sort of continue to sample collection and then to have the outcome data mature in this study. But this will be a definitive study, I believe, for demonstrating whether circulating tumor DNA at the time of diagnosis is prognostic in intermediate risk rhabdo. This study also had built in multiple serial samples along the way, so we'll also be able to understand how ctDNA levels change over time. This is just a snapshot of some data, very similar to our other data that we can detect ctDNA. We have a couple of examples where we found that ctDNA levels had risen very significantly at the time of relapse or progression. And what's fascinating again here is that when you look at the copy number plots, the top plot is from pre-treatment and the bottom is from a progression or relapse. The ctDNA levels are very similar in these two plots, but you can see brand new copy number changes at the relapse sample. And so we know that patients' tumors are changing over time on a genomic level. We also need to do some deeper sequencing of these diagnostic relapse pairs to see what point mutations are being gathered, whether new variant and new translocations are being acquired. But I think this is very compelling. And if we can start to show this kind of thing, this is just another example, across 20, 30, 40 patients, maybe we'll start to pick up recurrent patterns and understand some mechanisms that give rise to relapse disease. One more quick ctDNA story. This is very early days, but we're looking at circulating tumor DNA on the high-risk neuroblastoma study. In this cohort, ctDNA levels are extraordinarily high. In some blood samples, we're getting microgram or more of cell-free DNA out of the sample, and all of it is circulating tumor DNA. So it's as good as a biopsy sample for profiling the tumors. And we can see copy number changes in these variants. That's how we know we have high ctDNA levels. We can find MYC amplification. We can recapitulate across the cohort the common copy number variants that often happen and define neuroblastoma, high-risk neuroblastoma. And then we took a handful of these samples just as an exploratory pilot study and did deeper sequencing. And interestingly, in a couple of cases of patients who were enrolled in the study on the non-ALK arm, meaning an ALK mutation hadn't been found in their tumor, we actually could find an ALK mutation at a low allelic fraction in the plasma, suggesting that some subclones in some of these patients' tumors actually already have ALK variants at the time of diagnosis. And of course, it's totally unanswered whether patients who have these ALK variants will benefit from the addition of an ALK inhibitor to chemotherapy, and it's definitely unanswered whether patients who have subclonal ALK mutations would benefit from the ALK inhibitor. But this gives us a chance to start thinking about asking that question down the road. I think one of the amazing things is that now that we've sort of got some speed, blood samples are now being appropriately collected in Streck tubes that are ideal for liquid biopsy studies across many, many different trials now, with a big emphasis in the COG, sort of committing to serial collection of plasma samples in all the large phase 3 studies. NANT and other groups are also doing this. I'm going to skip over this section here, but just suffice it to say, we're also interested in early detection for patients with cancer predisposition, and the detection of early relapse in patients who have gone through therapy with the idea of trying to detect relapses early. And this is going to take some new technologies to try to improve our sensitivity for ctDNA detection. And there's a lot of approaches: ddPCR, patient-specific NGS panels, deeper genomes, fragmentomics, and even understanding chromatin changes by understanding how the cell-free DNA fragments line up with places where transcription factors bind in the genome. And all of that work is stuff that we're trying to be involved in at every level. We're interested in early detection in cancer predisposition syndromes such as Li-Fraumeni syndrome. Many of the technologies that I just glazed through extremely fast, we're thinking about how to apply to early detection in a patient we don't know has cancer, but is a very high risk of cancer. We know that Li-Fraumeni syndrome is really the most common cancer predisposition syndrome that gives rise to cancer in children. And in my clinic, I actually focus on cancer predisposition clinic in my outpatient clinic. So we're very interested in trying to find ways to detect cancer early. We know in LFS, Li-Fraumeni syndrome, that if we detect cancer early by using surveillance tools, that we can actually improve outcome. So this makes me quite optimistic that if we can apply liquid biopsies to this group, we may be able to further improve outcomes for this disease. We put together a multi-institutional consortium of cancer predisposition providers who are now collecting serial blood samples from patients with Li-Fraumeni syndrome, and honestly across a whole wide array of other cancer predisposition syndromes, with the hope that we can use those samples to really validate newer technologies for early cancer detection. And then just in the last couple of minutes, I wanted to talk about circulating tumor cells as another modality for liquid biopsy. So this is a little bit of a frame shift from what we just talked about. We've been thinking a lot about prognostication and adjusting therapy. I think CTCs are so hard to find and so hard to work with. I think it's very unlikely it's going to supplant the use of circulating tumor DNA for those sort of upfront prognostication and even for tracking disease and finding relapse. I think really cell-free DNA is going to be the workhorse there. But of course, there's a limitation to working with DNA. You can only find things that are related to DNA. You can't profile the tumor's RNA content or antigens like proteins and surface sugar modifications that sort of characterize different cancers. So we are looking at circulating tumor cells to try to understand those better. And we're thinking about it in the context of early phase trials. And the reason for that is that we know patients who are on early phase trials often have a higher burden of disease, so they're more likely to shed CTCs. And one of the big challenges of early phase trials that is yet to be solved is to develop a really great pharmacodynamic marker. When a patient goes in an early phase trial or an investigator opens an early phase trial and it works or it doesn't work, we often never know why. What happened in the course of that therapy affecting that tumor that made it work or made the tumors resistant, even when 100 out of 100 mice were cured with this therapy. Why is it that it completely failed to cure any actual patients who received the therapy? And honestly, because we can't do those end of therapy biopsies for those ethical reasons I mentioned earlier, this is a best chance to try to develop pharmacodynamic measurements of whether drugs that we think should work are hitting their target and whether those cancer cells are developing resistance mechanisms. So we're thinking about profiling RNA and antigens because a lot of the new evolving drugs that are interesting in the experimental therapeutics phase are either chromatin modifying agents like vorinostat or BRD4 inhibitors, or target surface antigens such as CAR T or antibody therapies, often bound to sort of a cytotoxic payload. And so in thinking about how to profile the transcriptome of CTCs at multiple time points early on in an early phase therapy, we've developed a workflow now where we take a blood sample, we enrich it for rigid big cells that characterize CTCs and try to wash away as many of the peripheral blood cells as we can. And then when we get out, we can then label with markers that are very specific for tumor cells and differentiate them from peripheral white blood cells, do flow cytometry, and then do things like PCR to look for variants we know. And this is just an example where we took a blood sample that didn't have cancer in it, spiked in some neuroblastoma cell lines, and were able to find the ALK variant that we knew characterized one of these neuroblastoma cell lines. We can also do a very similar experiment, again, a sort of a spike-in experiment with cell lines into blood. And after enrichment, instead of labeling them with antibodies and trying to sort out certain cells, just take that enriched sample where CTCs are still 1 to 1,000 basically, and put them into a 10x genomics experiment where we can do single cell on the whole population of cells that is left, and then use copy number profiles to pull out which of those subset of cells in that whole population of blood cells are CTCs. And in the spike-in experiment, you can see in the bottom right here that we're able to pull out the neuroblastoma cells by identifying the copy number profile from the gene expression data that characterize those that neuroblastoma cell line. And then very similar experiment. In this case, we took a patient sample, no spike in. This is a patient known to have high levels of neuroblastoma, who we had anticipated had high levels of neuroblastoma in their peripheral blood sample. We actually did the enrichment, did the labeling, and sorted individual neuroblastoma cells into wells, and then did a single cell RNA sequencing technology called SMART-seq2. And again, we're able to find that the cells we isolated were neuroblastoma cells, and the top shows the copy number profile inferred from the RNA-seq data, and on the bottom shows the ctDNA profile from that same patient. And although the boxes actually don't line up, if you match them 1-to-1, you can see that there's copy gains and copy losses in all the right places. So we are able to identify and profile these single cell CTCs out of blood samples from patients with neuroblastoma. In our proteomics work, we're taking a very similar approach. We do an enrichment, and then instead of labeling the antibodies with fluorescent antibodies, we label them with metal conjugated antibodies. And then instead of sorting them, we put them into a mass cytometer called the CyTOF machine. We profile all the cells that are in that size selection enriched blood sample. And then again, using the antigen pattern on the surface of cells, we're able to pull out which ones are cancer cells from peripheral blood cells. And in that antigen antibody metal conjugated cocktail, we include antibodies towards surface antigens that are being targeted by a therapy. So one of our ongoing studies is a trial that's using HER2 targeted therapy. And we're looking at we're finding the osteosarcoma cells in that cohort in those blood samples by the surface pattern of some of the antigens, and then measuring whether HER2 levels change over time when patients go on to the HER2 targeting therapy. We anticipate that HER2 levels will be downregulated in the context of this therapy and could be a mechanism of treatment resistance. We have 4 or 5 different studies now where we're looking at CTC work in this sort of pharmacodynamic way. And so lots more to come in this study. Tons of people to thank. These are highly collaborative projects. It's impossible to list all the people who contributed by writing a study and allowing us to get samples. Most importantly, well, funders are very important. We couldn't do this work without them, but most importantly, patients and families for contributing samples to this work. Stop now and take some questions. Thank you.
Wow, thank you so much. We have a few questions that have come in. I'm sure more are going to come in as we're answering these, but that was great. I'm going to start off with my own question first because I can. In predispositions, have you done any retrospective studies?
I don't know that of any retrospective serially collected blood samples from patients who have cancer predispositions. So we haven't been able to do that. The collections we're doing now are sort of being done simultaneous to assay development because we know we don't really have the tools yet to be sensitive enough as we want to be. So I guess by the time we actually do a lot of profiling of the samples we're collecting, it will be sort of retrospective. But it's also true that 15 years ago, we weren't doing as much genetic testing. So, now if we have a solid tumor patient at Dana-Farber, newly diagnosed patient with a solid tumor come in, everybody meets with a genetic counselor. Not everybody gets tested, but everybody meets a genetic counselor now. And so, we're finding a lot more cancer predispositions than we ever thought we would find. We're also now, because this is also happening in the adult levels, with adults who have cancer, we're meeting all these kids who have a 50% risk of inheriting a cancer predisposition from their father or mom who has an unusually young cancer, and who on their testing are found to have some sort of cancer predisposition syndrome. So, I don't know that it would have been possible to even collect these samples 20 years ago when we were doing, when we were finding who has a cancer predisposition so much less.
So, but for patients that have been followed for some years, would it be interesting to go back and see if they had blood and see, if things changed, if their cancer predisposition became cancer?
Yeah, and that's really the goal of this study, is to actually create that data bank, that blood sample bank. We're starting the minute we find out that they have a cancer predisposition syndrome at a time when we're actually sure they don't have cancer, and then collecting them at least every year, ideally every 6 months, for the rest of their lives. And in Li-Fraumeni syndrome, for example, it's about an 80 to 90% chance that at some point during their lifetime they'll develop cancer, and about 30% of patients who have Li-Fraumeni syndrome develop pediatric cancer. So, unfortunately, we're actually going to have over time a reasonable number of patients who do go on to develop cancer that we've been following for maybe years before they do, where we can look back and see how early can we look and see that they were developing cancer beforehand.
Okay, so Liz had a question, and she said, I think you mentioned at the start, but can you elaborate on whether this could be applied to brain tumors as is, or are there unique challenges for brain tumors?
I think the unique challenge is the blood-brain barrier. Our bodies were developed to protect our brains from things that might be in our blood. And so there's not a lot of crossover from brain tumors of liquid biopsy or cancer material being shed into the blood. But there are plenty of brain tumors where it's sort of routine to do, spinal taps to see if there's any evidence of cancer there. And I think that is probably a rich source of ctDNA in at least some brain tumors. There's a beautiful study out of St. Jude showing that ctDNA was definitely detectable in the CSF. I'm pretty sure it was patients with medulloblastoma in Cancer Cell not too long ago. I believe it was in Cancer Cell. Really great studies. So it's a matter of making sure you get the right fluid, but it's totally should be doable. And I think there are enough tools out there, as I sort of showed. The genomics can be applied the same way if you can find the sample that's going to give you enough cell-free DNA to interrogate.
Okay, I'm being told that if people raise their hand, we can unmute them and they can ask a question live. So we have that option. Meanwhile, I'm going to go back to the typed in questions. This one's from Ryan Schmidt, and it says, what variables determine the percent of ctDNA within a tumor type? And then it says disease burden, blood supply, mitotic activity.
Yes. I think we really don't actually totally know. My hypothesis, and this is a hypothesis, is that it's striking how much ctDNA is present in patients with newly diagnosed high-risk neuroblastoma. But that doesn't translate across the board to neuroblastoma broadly. So patients with localized disease don't necessarily have ctDNA levels that are all that much different than many of the other diseases we look at. So my hypothesis is that, we have a pretty strong sense that this is a disease where there's a lot of circulating cells. We get metastasis to the bone marrow, for example, in neuroblastoma. And so my hypothesis is that when cells are released into the blood, a large portion of those are not fit and they break down, and that's where we're getting the cell-free DNA. So I think that the tumor types where there's a high burden of metastasis happening, that the metastasis we're seeing is actually only a tip of the iceberg of the cells that are getting into circulation. The cells are getting into circulation lysine, and that's where we're getting our cell-free DNA. But it's a hypothesis. I think all the other variables that were listed in that question almost for sure contribute to ctDNA shed. We also see some genomic features that we associate with aggressive disease. Those patients tend to have higher levels too, although as you can see with that MyoD1 example, it's not 100% the case always.
I think it could be interesting. I don't know if you've ever treated a neuroblastoma patient who almost acts like a chronic neuroblastoma, where they have surges of sort of relapse and then it sort of settles down. It'd be interesting to sort of follow their ctDNA along those surges.
100%. I mean, I think the other thing we're also interested in is, in the context of neuroblastoma, is this stage 4M. I think it's renamed now. I'm not really, I don't take care of a lot of neuroblastoma patients in my own practice, but this phenomenon of high burden of disease, or even these small tumors that actually often go on to sort of never need treatment, but then some progress and do, like, could we actually differentiate that much quicker before patients have to develop symptoms or bulky disease? Could we figure out very early on who needs therapy or not? One of the challenges with that cohort is that we just haven't been able to get our hands on blood samples from that group of patients, because those studies are just hard to come by, and they didn't necessarily include blood sample collection, mostly because they were developed way before the advent of liquid biopsy. Nobody could have really anticipated it. So at some point, we absolutely need to do those studies, the one you mentioned, and I think this observation study as well.
I have two more questions, and if any more come in, because we don't have any more written ones. But one was, you mentioned in the clinical trials that they're collecting samples in the appropriate containers now.
Yep.
Are they actually going through and doing the testing, or are they just saving them for future?
Well, it depends. It's COG trials in particular are a complex beast, and there's often just enough funding from the NIH to run the trial itself. So the way this really works is that we're sort of, well, I've been trying my best with a lot of help from a lot of other people to be sort of a hound dog about making sure we're collecting really good samples that go into the COG repository. Then really it's kind of down to the investigators to sort of find funding to do the sequencing and then apply for the samples. And there's often sort of a lag time between when the sample's collected and when you can do the research. But there's also a lag time between when the study sort of finishes collecting and when the outcome data can be released on these trials. So it actually works out okay, but it's, like the reason I have like 1,000 different funders on my acknowledgement slide is because basically you need a new funder or a new grant for every single cohort, because these studies are actually pretty expensive. So, but that's basically how it's working. I think as we gain traction and as sort of the CCDI group sort of gets their feet down and gets more going from the NIH, I hope and I think there'll be more and more interest in thinking not just about tumor genomics and return of results of tumor genomics, but also the utilization of liquid biopsy to inform risk stratification. So, I mean, we've chatted sort of informally about whether there's opportunities there to sort of capitalize on how the NIH is really pushing forward, the use of genomic information to better treat patients with pediatric cancer. So I think we'll be able to capitalize on that.
And then my last question, and we're out of time anyways, is what is the cost to run one of these per patient?
ddPCR, which like only finds one variant, but it's very sensitive, can be as cheap as like you make an assay at about $300 for a patient, and you kind of can run it as many times as you want for no additional cost or very little additional cost. If we wanted to do 30x whole genomes on a blood sample, that's still $1,200 most places. So it kind of depends on the technology, and it's a wide range. Sequencing technologies the costs have continued to come down, and the use of single cell is just going to keep driving that, because there'll still be a need for a lot of sequencing, but within the same amount of money that people get from an NIH grant. So that's going to keep driving sequencing costs down. So I do think, we're going to get into a space where we can the next gen sequencing panels that we've developed, in a research environment cost us about $350 to run, $250 to run. So, we're going to get in a range where these things are affordable. We have to figure out how to make commercial providers offer them at an affordable rate. Foundation's test, for example, I think is $5,000 out of pocket per pop for their liquid biopsy test. And that's not their cost. That's way, way above their cost. So, right. I'm speculating, but yes.
Okay.
We can make this affordable. And reimbursable if we validate it, right? So that's, I think that's part of the key, is to make sure that we've validated this rigorously and from a statistical standpoint. And then, the next generation of studies will be to show that we can improve outcomes if we do better risk stratification. And then it'll be undeniable that that insurance companies will have to reimburse for this test because we can improve outcomes with it. And that's to me, the strategy.